लैसा भाई बोले थे कि क्लास 12 का एनसीईआरटी लाइन बाय लाइन लाने के लिए, ठीक। तो देखो रात के कितने 2:17 हो रहे हैं, 9 मार्च संडे। लेकिन देखो मैं सोना चाह रहा था लेकिन नींद नहीं आ रहा था, हो सकता है भाई का बोर्ड आ रहा हो। तो इसीलिए पहला जो एनसीईआरटी रीडिंग है यूनिट 6 का क्लास 12 का स्टार्ट जो हो रहा है, क्लास 12 का रिप्रोडक्शन करेंगे अभी तुरंत। और इसमें चैप्टर 1 रिप्रोडक्शन इन ऑर्गेनिज्म, ये तो डिलीट हो गया तो इसको नहीं करेंगे। बट सेक्सुअल रिप्रोडक्शन इन फ्लावरिंग प्लांट, चैप्टर 3 ह्यूमन रिप्रोडक्शन, चैप्टर 4 रिप्रोडक्टिव हेल्थ, ये तीनों चैप्टर करेंगे इस वीडियो में, वन शॉर्ट वीडियो में। और इसमें अपन क्या करेंगे, लाइन बाय लाइन रीड करेंगे पूरे, और कोशिश करेंगे कम से कम टाइम में हो जाए। ठीक है दोस्त, तो जिनके भी बोर्ड एग्जाम आ रहे हैं, नीट में भी यूजफुल है हर जगह, तो वीडियो देख सकते हैं। और भाई एक्सपेक्ट नहीं किया था, मतलब लोगों को अच्छा लगेगा, मतलब कमेंट देख के अच्छा लगा भाई, चलो स्टार्ट करते हैं। यूनिट 6 रिप्रोडक्शन, चैप्टर 1 रिप्रोडक्शन इन ऑर्गेनिज्म डिलीट हो गया, सेक्सुअल रिप्रोडक्शन पढ़ेंगे, उसके बाद ह्यूमन रिप्रोडक्शन करेंगे, उसके बाद रिप्रोडक्टिव हेल्थ करेंगे। सो बायोलॉजी इज एसेंस इन स्टोरी ऑफ लाइफ ऑन अर्थ, while individual organisms die without fail, species continue to live through millions of years unless threatened by natural or anthropogenic extinction. Reproduction becomes a vital process without which species cannot survive for long. Each individual leaves its progeny by asexual or sexual means. Sexual mode of reproduction enables creation of new variant so that survival advantage is enhanced. This unit examines the general principles underlying reproductive process in living organisms and then explain the details of this process in the flowering plants and humans as easy to relate representative examples. A relative perspective on human reproductive health and how reproductive ill health can be avoided is also presented to complete our understanding of biology of reproduction. Panchanan Maheshwari, 1904 to 1966. Born in November 1904 in Jaipur, Rajasthan, Panchanan Maheshwari rose to become one of the most distinguished botanists not only of India but of entire world. He moved to Allahabad for higher education, where he obtained his DSc during his college days. He was inspired by Dr. W. Dodigan, an American missionary teacher, to develop interest in botany and especially morphology. His teacher once expressed that if his student progress ahead of him, it will give him great satisfaction. These words encouraged Panchanan to inquire what he could do for his teacher in return. He worked on embryological aspect and popularized the use of embryological characters in taxonomy. He established the Department of Botany, University of Delhi, as an important center of research in embryology and tissue culture. He also emphasized the need for initiation of work on artificial culture of immature embryos. These days, tissue culture has become a landmark in science. His work on test tube fertilization and intra-ovarian pollination won worldwide acclaim. He was honored with Fellowship of Royal Society of London (FRS), Indian National Science Academy, and several other institutions of excellence. He encouraged general education and made a significant contribution to school education by his leadership in bringing out the first textbook of biology of higher secondary school published by NCERT in 1964. Chapter 2 Sexual Reproduction in Flowering Plants. So, 2.1 Flower of Fascinating Organ of Angiosperm. 2.2 Pre-fertilization Structures and Events. 2.3 Double fertilization. 2.4 Post-fertilization Structure and Events. 2.5 Apomixis and Polyembryony. Are we not lucky that plants reproduce sexually? The merits of flower that we enjoy gazing at, the scents and perfume that we swoon over, the rich color that attract us are all there as an aid of sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. A look at diversity of structure of the inflorescence, flowers and floral parts shows an amazing range of adaptation to ensure formal of the end product of sexual reproduction. The fruit and seeds. In this chapter, let us understand the morphology, structure, and the process of sexual reproduction in flowering plants and angiosperms. 2.1 Flowers, a fascinating organ of angiosperm. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are object of aesthetic, ornamental, social, religious, and cultural value. They have also been used symbols for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flower ornamental value that are commonly cultivated at homes and in gardens. Find out the names of 5 more flowers that are used in social culture celebration in your family. Have you heard of floriculture? What it does refer to? Or diagrammatic representation of longitudinal section of flower. So this is what? This is filament, this is what? Petal, and this is anther, and this is stigma, this is style, and this is sepal, and this is ovary. 2.2 Pre-fertilization structure and events. Much before the actual flower is seen on plants, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which led to the differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and female reproductive structure, the androecium and gynecium, differentiate and develop. You would recollect that androecium consists of whole stamens representing the male reproductive organs and gynecium represents the female reproductive organ. 2.1 Stamen microsporangium and pollen grain. Figure 2.2a shows the two parts of typical stamen, the long and slender stalk called filament and the terminal generally bilobed structure called anther. The proximal end of filament is attached to thalamus or petal of the flower. The number and length of stamens are variable in flower of different species. If you were to collect a stamen each from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagram would elucidate the range in shape and attachment of anther in different flowers. A typical angiosperm anther is bilobed, with each lobe having two theca. They are dithecous, often in figure 2.2b. Often longitudinal groups runs lengthwise separating the theca. Let's understand the various type of tissue and their organization in transverse section of an anther. Figure 2.23. The bilobed structure of an anther is very distinct in the transverse section of anther. The anther is four-sided tetragonal structure consisting of 4 microsporangia located at the corner of 2 in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of the anther and are packed with pollen grains. Structure of microsporangium in transverse section. A typical microsporangium appear near circular in outline. It is generally surrounded by 4 wall layers: the epidermis, endothecium, middle layer, and tapetum. The outer three wall layers perform the function of protection and help in dehiscence of anther to release pollens. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cells of tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think how tapetal cell could become binucleated? When the anther is young, a group of compactly arranged homogeneous cells called sporogenous tissue occupy the center of each microsporangium. Microsporogenesis. As the anther develops, the cell of sporogenous tissue undergo meiotic division to form microspore tetrad. What would be ploidy of the cells of the tetrad? A cell of sporogenous tissue is capable of giving rise to microspore tetrad. Each one is potential pollen of microspore mother cell. The process of formation of microspore from pollen mother cell PMC through meiosis is called microsporogenesis. The microspores, as they are formed, are arranged in cluster of cells, 4 cells. The microspore tetrad. As the anther mature and dehydrate, the microspore dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousand of microspore or pollen grains are formed that are released with dehiscence of anther. Pollen grains. The pollen grains represent the male gametophytes. If you touch and open the anther of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grain on your finger. Sprinkle these grains on drop of water taken on glass slide and observe under the microscope. You would rarely be amazed at variety of architectures, sizes, shape, colors, designs seen on the pollen grain. Different species figure in 2.4. तो पहले डायग्राम देख लेते हैं दोस्त. तो 2.2a और typical stamen, B 3-dimensional cut section of anther. So this is typical stamen and this is anther. This is what filament, also known as stalk, and these are pollen grains, and this is pollen sacs, and these are line of dehiscence. 2.3 or transverse section of young anther. So it is transverse section of young anther. This is connective tissue. This is connective, and this is epidermis, and this layer is endothecium, and then after this is tapetum. और tapetum के पहले क्या आता है? मिडल लेयर, उसका tapetum, then sporogenous tissue. And B enlarged view of one microsporangium showing wall layers. So enlarged view है क्या इस microsporangium के? सबसे बाहर क्या आ गया? epidermis, then endothecium, उसके बाद क्या आ रहा है? मिडल लेयर, then tapetum, उसके बाद क्या आ रहा है? माइक्रोस्पोर मदर सेल. A mature dehiscent anther, a mature dehiscent anther है, तो यहाँ पे ये सब pollen grains, and these are pollen sacs. Scanning electron microscope few pollen grains. तो कुछ pollen grain है माइक्रोस्कोप के अंदर, देखो अलग अलग डिजाइन वाले दिख रहे हैं ना. Pollen grains are generally spherical, measure about 25 to 50 micrometer in diameter. It has prominent two-layered wall. The hard outer layer called exine is made up of sporopollenin, which is one of the most resistant organic material known. It can withstand high temperature and strong acid and alkali. No enzyme that degrades sporopollenin is so far known. Pollen grain exine has prominent aperture called germ pores where the sporopollenin is absent. Pollen grains are well preserved as fossil because of presence of sporopollenin. The exine exhibit a fascinating array of pattern and design. Why do you think the exine should be hard? What is the function of germ pore? The inner wall of pollen grain is called intine. It is thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and generative cell. The vegetative cell is bigger, has abundant of food reserve and a large irregular shape nucleus. The generative cell is small and float in cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperm, pollen grain sets at two-celled stage. In the remaining species, the generative cell divide mitotically to give rise to male gametes before the pollen grain shed at three-celled stage. Figure 2.5. Or enlarged view of pollen grain tetrad B stage of microspore maturing into pollen grain. So this is pollen grain tetrad and this is maturing of pollen grain. So first stage, second and third, and here is four. तो पहले इस माइक्रोस्पोर है, मैच्योर हो रहा है पोलन ग्रेन में, तो ये यहाँ पे वाइक्यूअल है. देखो क्वेश्चन डायरेक्ट पूछ लेगा कि ये क्या है, तो ये दोनों वाइक्यूअल्स है, न्यूक्लियस है. एसिमेट्रिकल स्पिंडल. अब स्पिंडल फॉर्म क्या हो रहा है, सिमेट्रिकल या एसिमेट्रिकल. सो दिस इज सिमेट्रिकल स्पिंडल एंड दिस इज वेजिटेटिव सेल एंड दिस इज जनरेटिव सेल. तो देखो वेजिटेटिव सेल क्या है, बड़ा है, दिख ही रहा है बड़ा है. नेक्स्ट, pollen grain of his many species cause severe allergies and bronchial afflictions in some of people, often leading to chronic respiratory disorder, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as contaminant with imported wheat has become ubiquitous in occurrence and cause pollen allergy. Pollen grain are rich in nutrients. It has become a fashion in recent years to use pollen tablet as food supplements. In Western countries, a large number of pollen products in the form of tablets, syrups, etc. are available in market. Pollen consumption has become claimed to increase the performance of athletes in races horses. Pollen products. तो देखो यहाँ पे कुछ सिरप्स हैं, यहाँ पे pollen tablet 30 डेज तक लेने वाला टैबलेट है. ठीक, इस टैबलेट में कितना 30 टैबलेट आते हैं, ये भी आ सकता है ब्रो, कुछ भी हो सकता है. When once they said pollen grains have to land stigma before they lose viability, if they have to bring about fertilization, how long do you think the pollen grain retain viability? The period for which pollen grain remain viable is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and in some members of Rosaceae, Leguminaceae and Solanaceae, they maintain viability for months. You may have heard of storing semen, sperms of many animals including humans for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen at -196 degrees Celsius. Such stored pollen can be used in pollen banks similar to seed bank in crop breeding programs. 2.2.2 The pistil megasporangium or ovule and embryo sac. So, the gynosium represents the female reproductive part of flower. The gynosium may consist of single pistil, monocarpellary, or may have more than one pistil, multicarpellary. When there are more than one pistil may be fused together, syncarpus, or may be free apocarpus. Each pistil has three parts: the stigma, the style, and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated cylinder part beneath the stigma. The basal bulge part of pistil is called ovary. Inside the ovary, the ovarian cavity, locule. The placenta is located inside the ovarian cavity. Recall the definition of type of placenta that you have studied in class 11th. Arising from placenta are megasporangia, commonly called ovule. The number of ovules in an ovary may be one in wheat, paddy and mango, or many in papaya, watermelon and orchids. So यहाँ पर डायग्राम पहले देखते हैं. Figure 2.7. Or dissected flower hibiscus showing pistil, other floral part have been removed. So it is hibiscus का क्या है, hibiscus का ये pistil है, तो ये क्या हो गया, स्टिग्मा हो गया. And this is style, this is ovary and this is what, thalamus. B क्या है, मल्टी कार्पेलरी सीन कार्पस पिस्टिल ऑफ पेपेवर. तो यहाँ पे क्या हो रहा है, मल्टी कार्पेलरी मतलब कार्पेल बहुत ज़्यादा है और सीन कार्पस मतलब ज्वाइंटेड है. ठीक, इस किस में पाया जा रहा है, पेपेवर में पाया जा रहा है. एंड दिस इज व्हाट, दिस इज मल्टी कार्पेलरी एपोकार्पस गाइनोसियम मिचेलिया. ये मल्टी कार्पेलरी एपोकार्पस है लेकिन मतलब मीन्स ज्वाइंटेड नहीं है. एक इसमें पाया जा रहा है, मिचेलिया में पाया जाएगा. डी क्या है, डायग्रामेटिक व्यू ऑफ टाइपिकल एनाट्रोपस ओव्यूल. सो ये क्या है, हाइलम. व्हाट इज दिस, फ्यूनिकल ओव्यूल से ज्वाइंट होता है किस पार्ट पे, हाइलम पे. व्हाट इज दिस, दिस इज माइक्रोपाइलर एंड दिस इज माइक्रोपाइलर पोल. आउटर इंटीगुमेंट, ये इनर इंटीगुमेंट, और इसके अंदर क्या है, न्यूसिलस एंड दिस इज व्हाट, एम्ब्रियो सैक एंड दिस इज चैलेजल पोल. Chalazial pole is just opposite to the micropylar pole. The megasporangium ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to placenta by means of a stalk called funicle. The body of ovule fused with funicle in a region called hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has a one or two protective envelopes called integument. Integuments encircle the nucellus except at the tip where a small opening called micropyle is organized. Opposite the micropylar end, the chalaza representing the basal part of the ovule. Enclosed within integuments is a mass of cell called nucellus. Cells of nucellus have abundant reserve food materials. Located in the nucellus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from megaspore. Megasporogenesis. The process of formation of megaspore from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell (MMC) in micropylar region of nucellus. It is large cell containing dense cytoplasm and a prominent nucleus. The megaspore mother cell undergo meiotic division. What is the importance of megaspore mother cell undergoing meiosis? Meiosis results in the production of 4 megaspore. Figure 2.8a. So 2.8a, part of ovule showing large megaspore mother cell or dyad and tetrad of megaspore. So this is what, a large megaspore ovule है, megaspore show कर रहा है, megaspore mother cell, a micropylar end, this is nucellus, these are nucellus cell and this is megaspore mother cell. After division this form megaspore mother cell form कर रहा है megaspore dyad and this is nucellus and this is micropylar end, this is micropylar end and after फिर माइटोसिस उसमें हो रहा है तो 2 से 4 बना लिया मेगास्पोर टेट्राड. 248 न्यूक्लिएटेड स्टेज इन एम्ब्रियो सैक एंड मैच्योर एम्ब्रियो सैक और डायग्रामेटिक रिप्रेजेंटेशन ऑफ मैच्योर ओव्यूल. सो देखो मेगास्पोर टेट्राड बन गया ना, इसमें 3 जनरेट होकर एक फॉर्म करेगा तो 1 से 2 बना, 2 से 4 सेल, 4 से 8 बना. अब इसके बाद ये देखो, ये रहा माइक्रोपाइलर एंड पे सिनर्जिड ए बन रहा है. दिस इज एग, दीज आर दो सिनर्जिड एंड दिस इज पोलर न्यूक्लियाई, दिस इज सेंट्रल सेल एंड एक है एंटीपोडल सेल. अब इसका मैच्योर जो एम्ब्रियो सैक है वो कैसे है? तो ये है चैलेजल एंड, और चैलेजल एंड के अपोजिट माइक्रोपाइलर एंड। माइक्रोपाइलर एंड पे अपना क्या फिलिप फॉर्म apparatus है, और these are two synergid cells, and this is egg cell, this is central cell, and what is this is polar nuclei. So female gametophyte, in majority of flowering plant, one of the megaspore functional while other three degenerate. Only the functional megaspore develop into female gametophyte embryo sac. This method of embryo sac formation from single megaspore is termed as monosporic development. What will be the ploidy of cell of nucleus MMC, the functional megaspore and female gametophyte? Let us study the formation of embryo sac in a little more detail. In figure 2.8b, the nucleus of functional megaspore divide mitotically to form two nuclei which move to opposite pole, forming the two nucleated embryo sac. Two more sequential mitotic nuclear division result in the formation of four nucleated and later 8 nucleated stage of embryo sac. It is of interest to note that these mitotic division are strictly free nuclear. That is, nuclear division are not followed immediately by cell wall formation. After 8 nucleated stage, cells wall are laid down, leading to the organization of typical female gametophyte or embryo sac. Observe the distribution of cell inside the embryo sac in figure 2.8b, C. 6 of the 8 nuclei are surrounded by cell wall and organized into cell. The remaining two nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. There is characteristic distribution of cell within the embryo sac. 3 cells are grouped together at micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergid and 1 egg cell. These synergids have special cellular thickening at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tube into synergid. 3 cells are at the chelagel end and are called antipodals. The large central cell mentioned earlier has 2 polar nuclei. Thus, typical angiosperm embryo sac at maturity through 8 nucleated and 7 celled. 2.2.3 Pollination. In the preceding section, you have learned that the male and female gametes in flowering plant are produced in pollen grain and embryo sac, respectively. As both type of gametes are non-motile, they have to be brought together for fertilization to occur. How is this achieved? Pollination is mechanism to achieve this objective. Transfer of pollen grains sent from the anther to stigma of a pistil is termed as pollination. Flowering plants have evolved an amazing array of adaptations to achieve pollination. They make use of external agents to achieve pollination. Can you list the possible external agents? Kinds of pollination. Depending on the source of pollen, pollination can be divided into 3 types. First, autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from anther to stigma of the same flower. Figure 2.9. In a normal flower which opens and exposed to anther and stigma, complete autogamy is rather rare. Autogamy in such flower requires synchrony in pollen release and stigma receptive, and also the anther and stigma should lie close to each other so that self-pollination can occur. Some plants such as viola, common pansy, and oxalis and commelina produce 2 types of flowers: chasmogamous flowers, which are similar to flowers of other species with exposed anther and stigma, and clistogamous flowers which do not open at all. Figure 2.9. So, in such flower, the anther and stigma lie close to each other. When anther dehiscence in the flower bud, the pollen come into contact with the stigma to effect pollination. Thus, clistogamous flowers are invariably autogamous. There is no chance of cross-pollination landing on stigma. Clistogamous flower produce assured She said, even in absence of pollinators, do you think that Clistogamus is advantageous or disadvantageous to plant, and why? Figure 2.9, self-pollinated flower. This is self-pollinated flower, some beans, and cross-pollinated flower. This is sunflower cross-pollinated, and Clistogamus flower. This is Clistogamus. This is Chasmogamus and Clistogamus. Clistogamus is self-pollinated flower because it contains both male and female, and it is closed flower. Second, geitonogamy. Transfer of pollen grain from anther to stigma of another flower of the same plant. Although geitonogamy is functionally cross-pollination involving a pollinating agent, genetically it is similar to autogamy since the pollen grain come from same plant. Third, genogamy. Transfer of pollen grain from anther to stigma of a different plant. Figure 2.9. This is the only type of pollination which during pollination brings genetically different type of pollen to the stigma. Agents of pollination. Plants use two abiotic, wind and water, and one biotic animals agent to achieve pollination. Majority of plants use biotic agents for pollination. Only a small proportion of plants use abiotic agent. Pollen grains coming in contact with the stigma is a chance factor in both the wind and water pollination. To compensate for these uncertainties and associated loss of pollen grains, the flower produces enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by wind is more common amongst abiotic pollination. Wind pollination also required that pollen grains are light and non-sticky so that they can be transported in wind current. They often possess well-exposed stamens so that the pollen are easily dispersed into wind currents. Figure 2.10, and large often feathery stigma to easily trap airborne pollen grains. Wind pollinated flowers often have a single ovule in each ovary and numerous flowers packed into inflorescence. A familiar example is corn cob. Tassels you see are nothing but the stigma and style which wave in the wind to trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plants and limited to about 30 genera. Mostly monocotyl duns. As against this, you would recall that water is a regular mode of transport for male gametes among the flower plants groups such as algae, bryophytes, and pteridophytes. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need of water for transport of male gametes and fertilization. Some examples of water pollinated plants are Vallisneria and Hydrilla, which grow in fresh water, and several marine seagrasses such as Zostera. Not all aquatic use water for pollination. In majority of aquatic plants such as water hyacinth and water lily, the flower emerges above the level of water and pollinated by insect or wind as in most of the land plant. In Vallisneria, the female flower reach the surface of water by the long stalk, and male flower or pollen grain are released onto the surface of water. They are carried passively by water current. Figure 2.11a. Some of them eventually reach the female flower and the stigma. In another group of water pollinated plants such as seagrasses, female flowers remain submerged in water and the pollen grains are released inside the water. Pollen grains in many such species are long, ribbon-like, and they are carried passively inside water. Some of them reaches the stigma and achieve pollination. In most of the water pollinated species, pollen grains are protected from wetting by mucilaginous covering. Both wind and water pollinated flowers are very colorful and do not produce nectar. What would be reason for this? 2.10. A wind pollinated plant showing compact inflorescence and well exposed stamen. So this is wind pollinated plant. Majority of flowering plant use a range of animals as pollinating agents: bees, butterflies, beetles, wasps, ants, moth, birds, sunbirds, and hummingbirds, and bat are common pollinating agents. Figure 2.11b. Among the animals, insects, particularly bees, are dominant biotic pollinating agents. Even larger animals such as some primates, lemurs, arboreal tree-dwelling rodents, or even reptiles, gecko lizard and garden lizard, have also been reported as pollinators in some species. Often flowers of animals pollinated plants are specifically adapted for particular aspect of animals. Majority of the insect pollinated flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small, a large number of flowers are clustered into an inflorescence to make them conspicuous. Animals are attracted to flowers by color and or fragrance. The flowers pollinated by flies and beetles secrete fall odors to attract these animals. To sustain animals visit the flower have to provide rewards to animals. Nectar and pollen grains are useful in floral rewards. For harvesting the reward from the flower, the animal visitors come in contact with the anther and the stigma. The body of animal get a coating of pollen grains, which are generally sticky in animals' pollinated flowers. When the animal carrying pollen on its body come in contact with the stigma, it bring about pollination. A. Pollination by water in evaluation area. So this evaluation area and इसमें क्या हो रहा है, वाटर से pollination हो रहा है. This is female flower and this is stigma. This is male flower and this is female flower. Insect pollination. Yeah. In some species, floral rewards are providing safe place to lay eggs. As example is that tallest flower Amorphophallus. The flower itself is about 6 feet in height. A similar relationship exists between an species of moth and plant yucca, where both species, moth and plant, cannot complete their life cycle without each other. The moth deposits its eggs in the locule of ovary and the flower in turn get pollinated by moth. The larva of moth come out of egg as seed and start developing. Why do you observe some flower following plants or any other available to you? Cucumber, mango, people, coriander, papaya, onion, lobio, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out the animal visit them and whether they could be pollinators. You will have patiently observed flowers over a few days at different time of the day. You could also see whether there is any correlation characteristic of flower to animal that visit. Carefully observe if any visitors come in contact with the anther and stigma, as only visitors can bring about pollination. Many insects may consume pollen or nectar without bringing about pollination. Such floral visitors are referred as pollen nectar robbers or robbers. तो पॉलिनेशन भी नहीं कर रहा है और वहाँ से क्या कर रहा है, एक कंज्यूम भी कर ले रहा है पॉलिन को, उसको क्या नेक्टर रोबर बोलेंगे. यू मे or may not be able to identify pollinator, but you will surely enjoy your effort. Outbreeding devices. Majority of flowering plant produce hermaphrodite flowers, and pollen grain are generally to come in contact with stigma of the flower of the same flower. Continuous self-pollination result in inbreeding depression. Flowering plant have developed many devices to encourage self-pollination and to encourage cross-pollination, to discourage self-pollination and encourage cross-pollination. In some species, pollen release and stigma receptive are not synchronized. Either the pollen is released before stigma become receptive or stigma become receptive much before the release of pollen. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma ends of the same flower. Both these devices prevent autogamy. The third device to prevent inbreeding is self-incompatibility. This is genetic mechanism and prevents self-pollination from same flower or other flower of same plants from fertilizing the ovule by inhibiting pollen germination or pollen tube growth in pistil. Another device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on same plants such as castor and maize monoecious, it prevents autogamy but not geitonogamy. It's in several species such as papaya, male and female flower are present on different plants. That is, each plant is either male or female dioic. This condition prevents both autogamy and geitonogamy. Pollen pistil interaction. Pollination does not guarantee the transfer of right type of pollen. Incompatible pollen of the same species as the stigma. Often pollen of the wrong type either from the other species or from same plant, if it is self-incompatible, also land on stigma. The pistil has the ability to recognize the pollen whether it is right type compatible or wrong type incompatible. If it is right type, the pistil accepts the pollen and promote post-pollination event, that is, led to the fertilization. If the pollen is of a wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or pollen tube growth in style. The ability of pistil to recognize the pollen followed by its acceptance or rejectance in the result continuous dialogue between pollen grains and pistil. The dialogue is mediated by chemical components of the pollen interacting with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and interaction leading to the recognition followed by acceptance or rejection. As mentioned earlier, flowering compatibility pollination, the pollen grain germinate on stigma to produce pollen tube through one of the germ pore. The content of pollen grain move into the pollen tube. Pollen tube grow through tissue of the stigma and style entries to ovary. You would recall that in some plant pollen grains are set at two-celled condition of vegetative cell and generative cell. In such plant, the generative cell divide and form the two male gametes during growth of pollen tube in the stigma. In plant which set pollen in three-celled condition, pollen tube carry the two male gametes from beginning. Pollen tube after reaching the ovary enter the ovule through micropyle and then enter one of the synergid through filiform apparatus. Figure 2.212de. Many recent studies have shown filiform apparatus present at the micropylar part of synergid guide the entry of pollen tube. All these events from pollen deposition on stigma until pollen tube enter the ovule are together referred as pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by the promotion or inhibition of pollen. The knowledge gained in this area would help plant breeder in manipulating pollen pistil interaction, even in incompatible pollination to get desired hybrid. You can easily study pollen germination by dusting some pollen from flowers such as pea, chickpea, crotalaria, balsam and vinca on the glass slide containing drop sugar solution about 10%. After about 15 to 30 minutes, observe the slide under a low power lens in the microscope. You are likely seen pollen tube coming out of the pollen grain. As you shall learn that chapter on plant breeding in chapter 9, a breeder is interested in crossing different type of species and often gender to combine desirable character to produce commercially superior varieties. Artificial hybridization is one of the major approach of crop improvement program. In such crossing experiment, it is important to make sure that only the desired grains are used for pollination, and the stigma is protected from contamination from unwanted pollen. This is achieved by emasculation and bagging techniques. If the female parents bear bisexual flower, removal of anther from the flower bud before antherdescence using pair of forceps is necessary. This step referred to as emasculation. Emasculated flowers have to covered with bags suitable size, generally made up of butter paper, to prevent contamination of stigma with unwanted pollen. This process is called bagging. When the stigma bagged from attains receptivity, mature pollen grains collected from anther of the male parent are dusted on the stigma, and the flower are rebagged and the fruit allowed to develop. If the female parent produces unisexual flower, there is no need for emasculation. The female flower buds are bagged before the flower open. When the stigma becomes receptive, pollination is carried out using the desired pollen and flower rebagged. So here is what 2.12 of pollen grains germinating on stigma. बी एक pollen grain germinate हो रहा है किस पे स्टिग्मा पे. बी pollen tube growing through स्टाइल. ये pollen tube कैसे ग्रो कर रहा है स्टाइल में दिया. ये longitudinal section of pistil showing path of pollen tube growth. ये pistil का शो कर रहा है. देखो pollen tube है, यहाँ tube आया यहाँ से फिलीफॉर्म प्रेशर से वो सिनर्जिड में कैसे जा रहा है. ये pollen tube है, ये एंटीपोडल सेल्स शलाजल एंड, और ये क्या माइक्रोपाइलर एंड. सिनर्जिड माइक्रोपाइलर एंड में हो रहा है. ठीक, ये क्या हो गया, एग हो गया. दिस इज व्हाट पोलर न्यूक्लियाई. इनलार्ज व्यू ऑफ एन एग अपरेटस. डी का एनलार्ज व्यू है एग अपरेटस का. शोइंग एंट्री ऑफ पोलन ग्रेन इनटू सिनर्जिड. यहाँ पोलन ग्रेन एंट्री कर रहा है सिनर्जिड में. दिस दो एग क्या है मेल गैमेट एंड दिस इज व्हाट वेजिटेटिव न्यूक्लियस. ये क्या है फिलीफॉर्म अपरेटस. व्हाट इज दिस, दिस इज सिनर्जिड एंड दिस इज प्लाज्मा मेम्ब्रेन. ये सेंट्रल सेल, दिस इज एग न्यूक्लियस, दिस इज सेंट्रल न्यूक्लियस में होता है पोलर न्यूक्लियाई है. दिस इज पोलर न्यूक्लियाई. ये डिस्चार्ज हो गया क्या, स्पर्म डिस्चार्ज हुआ 1 into एग एंड 1 सेंट्रल सेल. सेंट्रल सेल से एक यहाँ पे डिस्चार्ज हुआ और एक न्यूक्लियाई से मेल गैमेट इससे डिफ्यूज हुआ, तो ये बना देगा इंडो स्पर्म न्यूक्लियस. तो ट्रिपल फ्यूजन में पढ़ेंगे. इसमें डबल फर्टिलाइजेशन 2.3 डबल फर्टिलाइजेशन. आफ्टर entering one of the synergid, the pollen release the two male gametes into cytoplasm of the synergid. One of the male gamete moves towards egg cell and fuses with nucleus, thus completing the syngamy. This results in the formation of diploid cell, the zygote. The other male gamete moves toward the two polar nuclei located in central cell and fuse with them to produce triploid primary endosperm nucleus. ठीक, PEN. As this involves fusion of 3 haploid nuclei, it is termed triple fusion. Since two type of fusion, syngamy and triple fusion, take place in embryo sac, phenomena is known as what? Double fertilization. An event unique to flowering plant. The central cell after triple fusion become primary endosperm nucleus PEC and develop into endosperm, while the zygote develop into what? Embryo. ठीक. So 2.10 A. Fertilization of embryo showing zygote and primary endosperm nuclei. These are generating synergid. This is zygote 2N and this is primary endosperm cell and this is triploid primary endosperm nucleus, and these are antipodal cells also degenerate. B. Stage of embryo development in dicot show reduced sizes compared to A. So B की ये सब क्या है, जाइगोट का फॉर्मेशन कैसे हुआ, 1 सेल से 2 सेल हो रहा है, कैसे 2 सेल से ग्लोब्यूलर एम्ब्रियो बन रहा है, ग्लोब्यूलर से हार्ट शेप बन रहा है, हार्ट शेप से ये मैच्योर हो रहा है. तो ये हार्ट शेप और मैच्योर दोनों में सस्पेंसर पाया जा रहा है. What is this? This is radical and this is cotyledon and this is what? plumule. ये मैच्योर हो गया तो डायरेक्ट नीट में आ जाएगा जाइगोट से ग्लोबुलर हेड, ग्लोबुलर एम्ब्रियो से हॉट शेप एम्ब्रियो एंड इससे प्लूम्यूल बन रहा है. 2.4 First, post fertilization structure and events. Following double fertilization event of endosperm and embryo development, maturation of ovule into seed and ovary into fruit are collectively termed as post fertilization event. 2.4.1 Endosperm. Endosperm develop precede embryo development. Why? The primary endosperm cells divide rapidly to form triploid endosperm tissue. The cells of this tissue are filled with reserved food material and are used for nutrition of developing embryo. The most common type of endosperm development, the primary endosperm nuclei undergo successive nuclear division to give rise to form free nuclear. This stage of endosperm development is called prenuclear endosperm. Subsequently, cell wall formation occur and the endosperm became cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei and surrounded by white kernel is the cellular endosperm. Endosperm may be either completely consumed by the developing embryo, that is pea groundnut bean, before seed maturation, or it may be precede matured seed, that is castor coconut, and be used during seed germination. Split open some seeds of castor, pea, beans, groundnut, fruit, and coconut and look the endosperm in each case. Find out whether the endosperm persistent in cereals, wheat, rice, and maize. Figure 2.14. A typical dicot embryo. B. Longitudinal section of an embryo grass. What is this? This is dicot embryo and this is monocot in present in plants, and this is dicot embryo. This is plumule. These are cotyledons. This is hypocotyle. This is radical. This is root cap. Sorry. Scutellum. This is coleopatile and this is shoot apex. This is epiblast and radical, and this is root cap. Radical covered by coleorhiza. Shoot apex covered by coleopatile. और scutellum क्या है? cotyledon है एक. 2.4.2 Embryo. Embryo develop at micropylar end of embryo sac where the zygote is situated. Most of the zygote divide only after certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to developing embryo. Though the seed differ greatly, the early stage of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. Figure 2.13 depicts the stage of embryogeny in dicotyledons embryo. The zygote gives rise to the proembryo and subsequently the globular, heart-shaped and matured embryo. A typical dicotyledons embryo, figure 2.14, consists of the embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledons is called epicotyle, which terminate with plumule or stem tip. This cylindrical portion of below the level of cotyledon is called hypocotyle, that terminate at the lower end in the radical and root tip. The root tip is covered with root cap. Embryo of monocotyledon, figure 2.14b, possess only one cotyledon. In the grass family, the cotyledon is called scutellum, that is situated toward one side lateral or of the embryo axis. It is the lower end. The embryonal axis has the radical or root cap enclosed in an undifferentiated seed called coleorhiza. The portion of embryonal axis above the level of attachment of scutellum is epicotyle. Epicotyle has a shoot apex and a few primordium enclosed in a hollow foliar structure, the coleoptile. Soak a few seeds in water, say of wheat, maize, pea, chickpea, groundnut overnight. Then split the seeds and observe the various parts of embryo in seeds. 2.4.3 Seeds. In angiosperms, the seed is final product of sexual reproduction. It is often described as fertilized ovule. Seeds are formed inside fruit. A seed typically consists of seed coat, cotyledon, and an embryonal axis. This cotyledon of embryo are simple structure, generally thick and swollen due to storage of food reserve, as in legumes. Mature seeds may be non-albuminous or ex-albuminous. Non-albuminous seeds have no residual endosperm, as it is completely consumed during embryo development, that is pea groundnut. Albuminous seed retain a part of endosperm, as it is not completely used up during embryo development, that is wheat, maize, barley, castor. Occasionally, in some seeds such as black pepper, beets, remnants of nucellus are also persistent. This residual persistent nucellus is the perisperm. Integuments of ovule harden as through protective seed coats. Figure 2.15. The micropyle remains a small pore in seed coat. This facilitates the entry of oxygen and water into seed during germination. If the seed matures, its water content is reduced and seed become relatively dry, 10 to 15% cell moisture by mass. The general metabolic activity of embryo slows down. The embryo may enter into a stage of inactivity called dormancy, or if favorable conditions are available, adequate moisture, oxygen, and suitable temperature, they germinate. And as ovule mature into seeds, the ovary develops into fruit. That is, the transformation of ovules into seed and ovary into fruit proceeds simultaneously. The wall of ovary develops into wall of fruit called pericarp. The fruit may be fleshy, as in guava, orange, mango, etc., or may be dry, as in groundnut and mustard, etc. Many fruits have evolved mechanism of dispersal of seeds. Recall the classification of fruits and their dispersal mechanism that you have studied in early classes. Is there any relationship between number of ovules in ovary and the number of seeds present in the fruit? The most common plants by time of fruit develops from ovary. Other floral parts degenerate and fall off. However, in few species such as apple, strawberry, cashew, etc., the thalamus also contribute the fruit formation. Such a fruit are false fruit. Most of the fruit, however, develops only from ovary are called true fruit. Although in most of the species, fruits are the result of fertilization. There are few in which fruit develop without fertilization. Such fruit are called parthenocarpic fruit. Banana is one of such example. Parthenocarpic can be induced through application of hormones such as such fruit are seedless. Figure 2.15 Structure of some seeds. Here are some structure of some seeds. So these are micropyle. This is cotyledon, and this seems to monocot seed. This is seed coat. This is endosperm. This is cotyledon, and this is hypocotyl root axis. This is shoot apical meristem, and this is root tip. B. False fruit, apple and strawberry. A में अभी ये भी है cotyledon, and this is seed coat. This is what shoot apical meristem. ये क्या है hypocotyl root axis. This is root tip, and this is endosperm. Pericarp. This is endosperm. This is scutellum. Coleoptile cover polymule, and coleorhiza cover radicle. This is radicle, and this is coleorhiza. False fruit or apple or strawberry. So these are false fruits made up of thalamus. So the is thalamus. These are seeds, and these are endocarp, and this is mesocarp. This is thalamus and achini. These contain seeds. Seeds offer several advantages of angiosperm. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seeds seed formation is more dependable. Also, seeds have better adaptive strategy for dispersal of new habitat and also species to colonize the areas. As they have sufficient food reserve, young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provide protective and young embryo. Being product of sexual reproduction, they generate new genetic combination leading to variation. Seed is the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for storage of seed, which can be used as food throughout the year and also be rice crop in next season. Can you imagine agriculture in absence of seed or in the presence of seeds which germinate straight away soon after formation and cannot be stored? How long do you think seed may remain after they disperse? This period again very different in few species of seed lose viability in few months. Seed of large number of species live for several years. Some seeds can remain alive for 100 years. These are several records very old yet viable. The oldest is that Lupinus arcticus excavated from Arctic to tundra. The seed germinated and flowered after the estimated record 10,000 year of dormancy. A recent record of 2,000 year of old viable seed is of date palm Phoenix dactylifera, discovered during the archaeological excavation of at King Herod Palace near Dead Sea. After completion of brief account of sexual reproduction of flowering plants, would be worth attempting to comprehend an enormous reproductive capacity of some flowering plant by asking the following question: how many eggs are presented in embryo sac? How many embryo sac are present in an ovule? How many ovules are present in ovary? How many ovaries are present in typical flower? How many flowers are present on a tree? And so on. Can you think some plant in which fruit contain very large number of seeds? Orchids fruit are one such category. Of each fruit contain thousands of tiny seeds. Similar in case of fruit of some parasite species such as Anabacae and Striga. Have you seen a tiny seeds of ficus? How large trees fica develop from that tiny seeds? How many billions of seeds does each ficus tree produce? Can you imagine any other example in which tiny structure can reproduce such large biomass over years? 2.5 apomixis and polyembryony. Although seeds in general are products of fertilization, a few flowering plants such as some species of Asteraceae and grasses have evolved a special mechanism to produce seeds without fertilization called apomixis. What is fruit production without fertilization called? Thus, apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways development of apomictic seed. In some species, the diploid egg cell is formed without reduction division and develop into the embryo without fertilization. More often, as in many citrus and mango varieties, some of nuclear cell surrounding embryo sac start dividing, protrude into embryo sac and develop into the embryo. In such species, each ovule contains many embryos. Occurrence of more than one embryo in a seed referred to as polyembryony. Take out some seed of orange and squeeze them. Observe how many number of the different sizes and shapes from each seed. Count the number of embryo in each seed. What would be genetic nature of apomictic embryo? Can they be called clones? Hybrid variety of several of our food vegetable crop are being extensively cultivated. Cultivation of hybrid has tremendously increased productivity. One of the problem of hybrid is that hybrid seeds have to be produced every year. If seeds collected from hybrid are sown, the plant in the progeny will segregate and do not maintain hybrid characters. Production of hybrid seeds is mostly costly, and hence cost of hybrid seeds become expensive for farmers. If these hybrids are made of into apoplectic, there is no segregation of character in hybrid progeny. Then farmer can keep the using hybrid seed to raise new crops year after year, and he does not have to buy hybrid seed year every year. Because of the important apomixes in hybrid seed, industry activity is going on in many laboratories about the world to understand the genetic apomixes and to transfer apomictic genes into hybrids. Summary: the flowers are sets of sexual reproduction in angiosperms. In flowers, endoecium consisting of stamens represent the male reproductive organ, and gynoecium consisting of female pistil represent female reproductive organs. Or typically, anther is bilobed, dithecous, and tetrasporangiate. Pollen grains develop inside sporangia. Four wall layers: the epidermis, endothecium, middle layer, and tapetum surrounded the microsporangium. Cells of the sporogenous tissue lying in center of the microsporangium undergo meiosis microsporogenesis to form tetrad or microspore. Individual microspore mature into pollen grains. Pollen grains represent the male gametophyte generation. The pollen grains have two layered wall, the outer exine and inner entine. The exine is made up of sporopollen and germ pore. Pollen grains may have two cells, vegetative cell and generative cell, or three celled or vegetative cell and two gametes, male gametes. At the time of shedding, the pistil has three parts: the stigma, the style, the ovary. Ovules are present in ovary. The ovules have a stalk called funicle, protective integument, and an opening called micropyle. The central cell is the nucellus, in which the archisporium differentiate. A cell of archisporium, the megaspore mother cell, divide meiotically, and one of the megaspore form embryo sac, the female gametophyte. The mature embryo sac 7 cells at nucleated at the micropylar end. The egg apparatus consisting of two synergids and an egg cell at the salagal end and three antipodals at the center of large central cell with two polar nuclei. Pollination is the mechanism to transfer pollen grain from the anther to stigma. Pollinating agents are either abiotic wind and water or biotic animals. Pollen pistil interaction involve all events from landing of pollen grains on stigma until the pollen grain enter into embryo sac, when the pollen is compatible, or inhibition when the pollen is incompatible. Following incompatible pollination, the pollen grain germinate on the stigma, and the resulting pollen tube grow through the style into ovule, finally discharged to male gamete in one of the synergids. Angiosperm exhibit dual fertilization because two fusion events occur in each embryo sac, namely syngamy and triple fusion. The product of these fusion are diploid zygote and triploid primary endosperm nucleus in the primary endosperm cell. Zygote develop into embryo and primary endosperm cell that develop endosperm tissue. Formation of endosperm always precedes development of embryo. The developing embryo passes through different stages such as pro-embryo, globular, and heart-shaped stages before maturation. Mature dicotyledonous embryo has two cotyledons and an embryonal axis with epicotyle and hypocotyle. Embryos of monocotyledons have a single cotyledon. After fertilization, ovary develops into fruit and ovule develop into flowers, into seeds. A phenomenon called apomixis is found in some angiosperms, particularly in grasses. It results in formation of seed without fertilization. Apomix have several advantages in horticulture and agriculture. Some angiosperms produce more than one embryo in food seed. The phenomena is called polyembryony. So सेक्सुअल रिप्रोडक्शन इन फ्लावरिंग प्लांट हम लोग का पूरा एनसीईआरटी लाइन बाय लाइन कवर हो गया है. ठीक, इसको जल्दी कवर करना हो जिनको 2x पे करके कर सकते हैं. बाकी 23 मार्च को लगभग है इसका बायोलॉजी का एग्ज़ाम 12th का बोर्ड का, वो लोग भी कर सकते हैं. तो अपना कंप्लीट हुआ भाई, तो कुछ ब्रेक लेते हैं, उसके बाद स्टार्ट करेंगे अपना ये क्या ह्यूमन रिप्रोडक्शन चैप्टर 2 मान लो, बिकॉज पहला थोड़ी है ना ह्यूमन रिप्रोडक्शन. तो 3:27 to 3:33 AM ब्रेक. आते हैं भाई शॉर्ट ब्रेक के बाद. तब तक आप लोग थोड़ा थोड़ा मूड फ्रेश कर लीजिए, उसके बाद फिर करते हैं रिस्टार्ट इसको. दिल की रान क्या बोलता है भाई, रानू दिल्ली की नहीं मुंबई की रानू है ना भाई. pollen and pistil components and interaction leading to the recognition followed by acceptance or rejection. तो भाई चलते हैं भाई, स्टार्ट करते हैं। हो गया रेस्ट कंप्लीट। तो करने से पहले एक कुछ छोटा मोटा मोटिवेशन सुन लो, आगे एनर्जी मिल जाएगा। तो कुछ ऐसा है, जीतेंगे हम ये वादा करो। क्या? जीतेंगे हम ये वादा करो, कोशिश हमेशा ज़्यादा करो। किस्मत भी रूठे पर हिम्मत ना टूटे, किस्मत भी रूठे पर हिम्मत ना टूटे, मजबूत इतना इरादा करो। तो भाई मजबूत इरादा अपना इतना करो कि किस्मत रूठ जाए, बस तुम्हारा हार्डवर्क करते रहो भाई, किस्मत के भरोसे ना रहो। चलो अपना टाइम हो गया ब्रो, स्टार्ट करो, लेट्स गो। तो भाई रेस्ट हो गया ना आप लोगों का। चलिए अब स्टार्ट करते हैं ह्यूमन रिप्रोडक्शन। तो भाई यही चैप्टर ना क्लास 8 में ही करते थे लोग बेंच के पीछे, बेंच पे। लेकिन भाई बहुत ही क्या है, चैप्टर बहुत इम्पोर्टेंट है भाई, इसको एक सीरियस नोट पे हम लोगों को पढ़ना चाहिए। इसमें कुछ भी हंसने या वैसा बात नहीं है यार, पूरा हम लोग का समझ लो ह्यूमंस का अस्तित्व, मतलब कैसे हो रहा है डेवलपमेंट, हर एक चीज़। तो ये बहुत क्रूशियल टॉपिक है, इसलिए इसको सीरियस होकर हम लोग को समझना चाहिए या पढ़ना चाहिए। चलिए रीड करेंगे हम लोग, समझेंगे क्या, मतलब रीड करेंगे उसमें समझ भी जाएंगे, मतलब एक्सप्लेन इसमें नहीं हो रहा है ना। 3.1 द मेल रिप्रोडक्टिव सिस्टम, 3.2 पढ़ेंगे द फीमेल रिप्रोडक्टिव सिस्टम, 3.3 गैमेटोजेनेसिस, 3.4 menstrual cycle, 3.5 fertilization and implantation, 3.6 fertilization and embryonic development, 3.7 parturition and lactation. As you are aware, humans are sexually reproducing and viviparous. The reproductive event in humans include formation of gametes, gametogenesis, that is sperm in males and ovum in females, transfer of sperm into female genital tract, insemination and fusion of male and female gametes, fertilization leading to formation of zygote. This is followed by formation of development of blastocyst and its attachment to uterine wall, implantation, embryonic development, gestation and delivery of the baby, parturition. You have learned that these reproductive events occur after puberty. There are remarkable differences between the reproductive events in the male and in females. For example, sperm formation continues even in old men, but formation of ovum ceases in women around age of 50 years. Let us examine the male and female reproductive system in humans. 3.1 The male reproductive system. The male reproductive system is located in the pelvis region, figure 3.1a. It includes a pair of testes along with accessory ducts, gland and external genitalia. So 3.1 diagrammatic sectional view of male pelvis region. This is what ureter, and this is seminal vesicle, and this is urinary bladder. And what is this? There was this is vas deferens, and this is prostate gland, and this is penis, this is urethra, and this is glans penis. And what is this? This is foreskin that cover glans penis. This is testes, and its scrotum pouch, and this is bulbourethral gland, and this is anus, and this is rectum, and this is what? Ejaculatory duct. The testis is situated outside the abdominal cavity within pouch called scrotum. The scrotum helps in the maintaining of low temperature of testes, 2 to 2.5 degrees Celsius lower than the normal internal body temperature necessary for the spermatogenesis. In adults, each testis is oval in shape with a length of about 4 to 5 centimeter and a width of 2 to 3 centimeter. The testis is covered by a dense covering. Each testis has about 250 compartments called testicular lobules in figure 3.1b. Each lobule contains 1 to 3 highly coiled seminiferous tubules. 1 to 3 highly coiled seminiferous tubules in which sperm are produced. Each seminiferous tubule is lined on inside by two types of cells called male germ cells, spermatogonia and Sertoli cells. Figure 3.2. The female germ cells undergo meiotic division, finally leading to sperm formation, while Sertoli cell undergo nutrition to the germ cells. The region outside the seminiferous tubule called intercellular space, interstitial space, contain small blood vessels and interstitial cell or Leydig cells. Leydig cells synthesize and secrete testicular hormone called androgen. Other immunologically competent cells are also present. The male sex accessory duct include rete testi, vasa efferentia, epididymis and vas deferens in figure 3.1b. The seminiferous tubules of testes open into vasa efferentia through rete testis. The vasa efferentia leaves the testis and open into epididymis located along posterior surface of each testis. The epididymis lead to the vas deferens that ascend to the abdomen and loops over the urinary bladder. It receives the duct from seminal vesicles and open into urethra as ejaculatory duct. These ducts store and transport sperm from testes to outside the body through urethra. The urethra originates from urinary bladder and extends through penis to external opening called urethral meatus. Figure 3.1b, diagrammatic view of male reproductive system, part of testes open into in detail. So this is foreskin, this is glans penis, and these are testicular lobules, and this is rete testis, and this vasa efferentia connect rete testis to epididymis, and this epididymis later joined to vas deferens, and vas deferens joined to joined to prostate gland, and this is ureter, this is urinary bladder, this is seminal vesicle, and this is prostate gland, this is bulbourethral gland, and this is urethra, this is testis. The penis of male external genitalia, it is made up of special tissue help in erection of penis, facilitate insemination. The enlarged end of penis called glans penis is covered by loose fold of skin called foreskin. The male accessory gland, figure 3.1ab, include paired seminal vesicles of prostate and paired bulbourethral gland. Secretion of these glands constitutes seminal plasma, which is rich in fructose, calcium and certain enzymes. The secretion of bulbourethral gland also help in lubrication of penis. So यहाँ पे fructose है, calcium and certain enzymes. So here is diagrammatic section of view of seminal vesicle. This is interstitial cell, and these are spermatogonia. This is spermatozoa, and this is what? This is Sertoli cell, and this sectional view of seminiferous tubule. 3.2, the female reproductive system. The female reproductive system consists of a pair of ovaries along with a pair of oviduct, uterus, cervix, vagina and external genitalia located in the pelvic region. Figure 3.3a. This part of system along with a pair of the mammary glands are integrated structurally and functionally to support the process of ovulation, fertilization, pregnancy, birth and child care. Ovaries are the primary female sex organs that produce female gametes, ovum and several estroidal hormones, ovarian hormones. The ovaries are located one of the each side of lower abdomen. Figure 3.3b. Each ovary is about 2 to 4 cm in length and connected to pelvic wall and uterus by ligaments. Each ovary is covered by a thin epithelium cell encloses the ovarian stroma. The stroma is divided into two zones of peripheral cortex and inner medulla. 3.3 diagrammatic section view of female pelvis showing reproductive system. This is what? This is cervix, this is rectum, this is vagina, this is anus, and this is what? Uterus, this is urinary bladder, and this is pubic symphysis, this is what? Urethra, and here is clitoris, and here is labia minora, this is what? Labia minora or a labia majora, and this is what? Vaginal or orifice. The oviduct or fallopian tubes, uterus and vagina constitute the female accessory duct. Each fallopian tube about 10 to 12 cm long and extend from periphery of each ovary to uterus. The part closer to ovary is the funnel-shaped infundibulum. The edge of the infundibulum possesses finger-like projection called fimbriae, which help in the collection of ovum after ovulation. The infundibulum leads to the wider part of oviduct called ampulla. The last part of oviduct, isthmus, has narrow lumen and it's joined to uterus. The uterus is a single and it is called omb. The shape of uterus is like inverted pear. It is supported by ligaments attached to pelvic wall. The uterus opens into vagina through narrow cervix. The cavity of cervix is called cervical canal. Figure 3.3b, which along with vagina form birth canal. So birth canal contains both cervical canal plus vaginal canal. The wall of uterus has 3 layers of tissue. The external thin membrane perimetrium, middle thick layer is smooth muscle myometrium, and inner glandular layer endometrium that lined by uterine cavity. The endometrium undergoes cyclic changes during menstrual cycle, while the myometrium exhibits strong contraction during delivery of baby. The female external genitalia includes mons pubis, labia majora, labia minora, hymen, and clitoris. Mons pubis is a cushion of fatty tissue covered by skin and pubis hair. The labia majora are fleshy fold of tissue which extend down from mons pubis and surrounded the vaginal opening. The labia minora and paired fold of tissue under labia majora. The opening of vagina is often covered partially by membrane called hymen. The clitoris is tiny finger-like structure which lies at the upper junction of two labia minora above the urethral opening. The hymen is often torn during the first coitus intercourse. However, it can also be broken by sudden fall, jolts, insertion, and vaginal tampon. Active participation in some sports like horseback riding, cycling, etc. In some women, the hymen persists even after coitus. In fact, presence and absence of hymen is not a reliable indicator of virginity or sexual experience. So, figure 3.3 diagrammatic sectional view of female reproductive system. This is uterine fundus. This is what? Uterine cavity. This is endometrium, and this is myometrium, and this is what? Perimetrium. And this is cervix. This is cervical canal. This is vagina. This is ovary. This is fimbriae. This is infundibulum. This is ampulla, and this is isthmus, and this is collectively called fallopian tube. Mammary gland. A functional mammary gland is characteristic of all female mammals. The mammary gland are paired structures breast that contain glandular tissue and a variable amount of fats. The glandular tissue of each breast is divided into 15 to 20 mammary lobes containing clusters of cells called alveoli. The cells of alveoli secrete milk, which is stored in cavities of lumen of alveoli. The alveoli open into mammary tubules. The tubules of each lobe join to form mammary duct. Several mammary ducts join to form voidal mammary ampulla, which is connected to lactiferous duct through which milk is sucked out. So it is what? The diagrammatic sectional view of mammary gland. So this is rib. These are muscles between ribs, and this is pectoralis major muscles. तो सबसे बाहर अंदर क्या है? या फैट है. फैट से कहाँ जा रहा है? मेमरी लोब में मिल्क फॉर्मेशन होके जा रहा है. यहाँ से कहाँ जा रहा है? मेमरी एल्वुलस में जा रहा है. मेमरी एल्वुलस से होके मेमरी डक्ट में, मेमरी डक्ट से एम्प्यूला रीजन, एंड एम्प्यूला से होके लैक्टिफेरस डक्ट में, लैक्टिफेरस डक्ट से निप्पल्स में, एंड दीज आर व्हाट? एरियोला. So, 3.3 gametogenesis. The primary sex organ, the testes in the males and the ovary in the females, produce gametes, that is sperm and ovum, respectively, by the process called gametogenesis. In testes, the immature male germ cell spermatogonia produce sperm by spermatogenesis that begin at the puberty. The spermatogonia, singular spermatogonium present on the inside wall of seminiferous tubule, multiply by mitotic division and increase numbers. Each spermatogonia is diploid and contains 46 chromosomes. Some of the spermatogonia, called primary spermatocytes, periodically undergo meiosis. Primary spermatocytes complete the first meiotic division, reduction division, leading to the formation of two equal haploid cells called secondary spermatocytes, which has also 23 chromosomes each. This secondary spermatocyte undergoes a secondary meiotic division to produce 4 equal haploid spermatids. Figure 3.5. What would be the number of chromosomes in the spermatids? The spermatids are transformed into spermatozoa sperm by process called spermiogenesis. After spermiogenesis, sperm has become embedded in the Sertoli cell and finally released from seminiferous tubule by process called spermiation. Spermatogenesis starts at the age of puberty due to significant increase in the secretion of gonadotropin releasing hormone GnRH. This, if you recall, is a hypothalamic hormone. This increased level of GnRH then acts on anterior pituitary and stimulates secretion of two gonadotropins, luteinizing hormone LH and follicle stimulating hormone FSH. Luteinizing hormone LH acts on Leydig cell and stimulates synthesis and secretion of androgen. Androgen in turn stimulates the process of spermatogenesis. Follicle stimulating hormone acts on Sertoli cell and stimulates secretion of some factors which help in the process of spermiogenesis. Let us examine the structure of sperm. It is a microscopic structure composed of head, neck, middle piece and tail. A plasma membrane envelops the whole body of sperm. The sperm head contains an elongated haploid nucleus, the anterior portion of which is covered by cap-like structure acrosome. The acrosome is filled with enzymes that help in fertilization of ovum. The middle piece possesses numerous mitochondria which produce energy of movement of tail that facilitate sperm motility, essential for fertilization. The human male ejaculates about 200 to 300 million sperms during a coitus, of which for normal fertility at least 60% of his sperms must have normal shapes and size, and at least 40% of them show vigorous motility. Sperms released from seminiferous tubules are then transported by accessory duct. Secretion of epididymis, vas deferens, seminal vesicles and prostate are essential for maturation and motility of sperm. This seminal plasma along with sperms constitutes semen. The functions of male sex accessory duct and glands are maintained by testicular hormone androgen. Diagram of sectional view of seminiferous tubule. This is spermatozoa, this is spermatids, this is secondary spermatocytes, and it is primary spermatocyte and Sertoli cell, and here is spermatogonia. Structure of sperm. This is head, the plasma membrane, here is acrosome, this is nucleus containing chromosomal material, this is neck, this is middle piece, and these are mitochondria, energy rich source for swimming. This is tail. The process of formation of mature female gamete is called oogenesis, which is markedly different from spermatogenesis. Oogenesis is initiated during embryonic development stage when a couple of million of gamete mother cell oogonia are formed within each fetal ovary. No more oogonia are formed and added after birth. These cells start division and enter into prophase of meiotic division and get temporarily arrested at the stage that primary oocyte. Each primary oocyte then gets surrounded by a layer of granulosa cell and is called primary follicles. Figure 3.7. A large number of these follicles degenerate during phase of from birth to puberty. Therefore, in puberty, 60,000 to 80,000 primary follicles are left in each ovary. The primary follicle gets surrounded by more layers of granulosa cell and new thega and are called secondary follicles. These secondary follicles soon transforms into tertiary follicles, which is characterized by fluid-filled cavity called antrum. The theca layer is organized into an inner theca interna and outer theca externa. It is important to draw your attention that it is this stage that primary oocyte within tertiary follicle grow in size and completes first meiotic division. It is an unequal division resulting in the formation of a large haploid secondary oocyte and a tiny first polar body. 3.8. The secondary oocyte retain bulk of the nutrient-rich cytoplasm of the primary oocyte. Can you think of any advantage of this? Does the first polar body born out first meiotic division divide further or degenerate? At present, we are not very certain about this. The tertiary follicle further changes into mature follicle or Graafian follicle. Figure 3.7. These secondary oocytes form a new membrane called zona pellucida surrounding it. The Graafian follicle now rupture to release secondary oocyte ovum from the ovary by process called ovulation. Can you identify major difference between spermatogenesis and oogenesis? A diagrammatic representation of spermatogenesis and oogenesis given in 3.8. 3.7 diagrammatic sectional view of ovary. Here are blood vessel and this is primary follicle, this is tertiary follicle or antrum, and this is Graafian follicle, this is ovum, and these are corpus luteum. Schematic representation of